From Microbe TV, this may be office hours for Wednesday, April 17th, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet. 5 minutes before I was about to go live, the internet at the incubator went down, and it's still down. So I tethered it to my iPhone. And it's amazing that it is working. The only thing is I don't have a laptop to monitor the chat. Hold on. Let's see. I got a second iPhone here I can use to set that up, right? Swipe up to get started. Okay. Where's the bloody, where is the hotspot here? It's in settings. Okay, all these things. Okay, where's the hotspot? Hotspot. Where's the hotspot? We don't want Wi-Fi on. We want the hotspot. Sorry, folks, to involve you in this sausage making. I'm looking for the hotspot on this phone because I want to connect my laptop up to it. Hotspot. Oh, come on. I search for hotspot and it just gives me crap. Spot, S-P-O-T, hotspot, cellular. Maybe it's under cellular. There we go. Cellular personal hotspot. Turn it on. Right. Bluetooth and USB only. What? I'll put it on Wi-Fi. Okay. And here's the password. Very good. I'm almost there. And then I can see hotspot settings, personal hotspot. Thank you, Steph. It's called iPhone. Yeah. Let's type in the password. Why didn't it ask me for a password? Huh? Let's see if I got internets. Cellular, yeah. Oh, maybe it's going to work. It is. It's working on the hotspot. Okay, folks, welcome. It looks like we're going to be in business here. At least I have some broadcast. And welcome, everybody. Who is here tonight? Tell me where you're from while I'm going to thank the moderators here tonight. Let's see. I have to scroll down because this thing isn't loading. It's slow. Why isn't it loading? iPhone. Let's see who's here. I know that here's Andrew here. Welcome, Andrew. Tom is here. Vanity Nutrition is here. Les is here. And I believe that's it for tonight for moderators. Thank you all for waiting. It's only 13 minutes. So the internet is out, but I'm connecting for the stream through my iPhone. And then I tried to connect to my other laptop to see the actual stream, but it's not showing up. I don't know why. It should. It should. Maybe, is there an issue there? Anyway, we won't worry about that. We'll just let the ball spin. And welcome, everybody, to Office Hours. I have a little mini lecture for you tonight. And let's see where people are from. I can start. I'm not a genius. Oh, I got the thing here now. I got the stream separately. I didn't think a hotspot would work. We have pretty good cell signal here. I didn't think it would work, but it's working. I got you guys on the picture, and I got my thing here. And I'm not going to check my internets. It's not in yet. No. Let's quit that. I guess I don't want to check it because I have two things running and I don't want to interrupt it. So we're going to have to go on cell phone tethering. It's fine. So I'm glad to be able to do this because I was all ready, and I don't want to have to cancel it. So let's see where everyone is from. Angry Penguin is from Cold, Wet Toronto. Notice we have the white fonts. We're back to the white fonts. Let's see who else is here. Lulu is waiting. Where are you from? It wasn't a proof that the incubator is a hotspot. Walter is from North Central Massachusetts. Our favorite sausage. Yeah, I'm a sausage head. Ay. Thank you for helping me, everyone. And yes, we're a year out of Epitope 1000. It was April 15th, just 2 days ago. Stinking tax day. Oh, my God, I hate tax day. And it was a year ago. It was the 15th of April. It was a Saturday, so you didn't have to have your taxes until Monday. But, no, Karen is not around. Karen's not feeling good tonight, unfortunately. But I did talk to her, and she suggested using the hotspot. So thank you. Krishnan, all right, from Malaysia. That's far away. I was just talking about Malaysia today in class. They were the first Nipah virus outbreak in the Perak district of Malaysia. James in Austin, Texas. Hello, James. Rima is in Iowa. Silent Runner is in Kansas. John is in Minneapolis. Elizabeth is in West Virginia. Abdur Rahman Magdi is in New York City tonight. Elisio is from the Amazon region. Oh, my. Always raining hot. So I've learned that you put extra letters, last letters on, on these words to emphasize them. Like, yes, hot. Claire is from the UK. Hunter is in Wisconsin. Jan is in Tucson, Arizona. 5G to the rescue. Stinking wired internet. Stinking wired internet. But why does it have to go out? Why? It should be perfect. Seychelles, British Columbia is Lucy. Pete is from London. Shut your internet down and restart. Well, I can't do anything now because the internet's in the closet. And unless I go look at the light and see if it's green, I won't know. But I will give it 10 minutes or so, and I'll go look. I suspect it won't come back till tomorrow, right? Who's going to work on it tonight? EHS Talent from Eastern Pennsylvania. Aman is from near D.C. Doreen is in Hartford, Connecticut. Marie is in Dolton, Illinois. Bob is from Winnipeg. Yeah. If it's working as is, don't fix it. Hard-earned engineer knowledge. Yes. If it ain't broke, don't fix it because you'll break it. Abilash is in Frankfurt, Germany. And Lori is in Charlottesville, Virginia. Yeah, you need to unplug the modem and plug it in. Yeah, I'd done that before, but I'm not going to do it again until we're some ways in, and I have some confidence that the internet is up. So actually, Jan, you can look on your app. Jan has helped me with the internet in the past, and she has an app on her phone that can look at it. So if you periodically look, you tell me if it comes back up, and then we'll switch back to it. Check if the bill got paid. Yeah, it's an automatic billing thing, and our card is not expired. It should be okay. Yeah, but that would be a silly thing to do. Mood Indigo is from India. What a lovely name, Mood Indigo. I really like that. So welcome, everyone. It's back up. So what should I do? Should I go to it? Let's see. You know, the one, let's try it. incubator. Let me go look at the light. Hold on. Yeah, it is back up. Now I don't want to switch it because everything's fine, right? So screw it. But thank you, Jan, for that. And if anything goes wrong with the phones are plugged in, so it should be okay. Keep it as is, yeah. If it ain't broke, don't fix it. Who knows? It could go out again. Anyway, folks, I want to do a little mini lecture. I have an interesting topic tonight that I thought you would be interested in. As, of course, in my horror at this internet business, I didn't queue up the presentation. So let me do that now. We'll go to the office hours folder, and we will go to 417. So now let me remember how I did this, because I don't remember. No, that wasn't it. How did I do this? I know I did the screen share. Let's take Mood Indigo off here. Sorry, I like your colors, but we need to get you off. It's really small, but let's turn on the screen share. Oh, come on. All right, screen share is on. But it doesn't look like it's sharing anything, right? I have no idea. Ecamm Live, no, it's not sharing it. But we also want to put Vinny in here. You know, the problem with technology is that if you don't use it, you lose it, right? If you don't use it, you lose it. So let's put that back on top of the screen share. I did. Why isn't it showing up there? Now you see it's not showing up in the right screen. So let's pull it over here and see if it's pulled up there. I know it's blank screen. It'll work. Don't worry about it. It'll work. There you go. See? It slides in. Let me just make this a little bigger. Let's try 75%. I think that's good. We'll take off the formatting bar here. Let's see. Slide only. We'll do that. I think that'll do it. So what I want to do, and I think you can see me. Yeah, I'm in the upper left there. Good. I want to talk a little bit about how viruses cause cancer, very briefly, just like 20 minutes or so. We did this lecture a couple of weeks ago, last week, and I really think it's a cool lecture, so let's do that. Now, how do I get to the next freaking slide? I don't have the navigator here. I'm not going to hit play, no. I guess I need to have the navigator here on the left so I can, because I'm not going in full screen mode. Although I guess I could. Let's try it. Is there a Vinny there? I don't think so. I think there is, yeah. I think there's still a Vinny. So it says a 30-second or so lag on my monitor. Yeah, there it is. Good. Just transformation and oncogenesis. They're two separate things, okay? It's not the same. So let's see what transformation is. Now I don't have, there's my pointer. Let's do an experiment to demonstrate transformation. So you take a hamster embryo and you chop it up and you separate all the cells singly by treating with trypsin. And then you put them on plates. You have two plates and you have one that doesn't get any treatment. And these cells will grow as a lovely monolayer, we call it. On the right here, you see the normal cells. They're nice and flat. And eventually they will fill the dish and then they stop dividing. But if you treat the cells with a mutagen, a chemical that induces mutations, some of the cells will become transformed and they form colonies that are aberrant. And you can see them on the right here. They are piling up on one another. They don't respect each other's boundaries. That's what transformation is. It's a change in the properties of the cell caused by mutations in the genome. It is not cancer. It is just different properties. So the cells become immortal. They will grow forever like HeLa cells. So HeLa cells were taken from Henrietta Lacks. She had a cervical tumor, and they removed a piece of it, and it grew in culture. And that's because the virus, human papillomavirus, had integrated into the genome of her cancer cells and made them divide and become a cancer. So the first thing that the virus did was to transform the cells, made them immortal. They lose anchorage dependence, so you don't need to be attached to a plastic anymore. They don't stop growing when they touch each other, the loss of contact inhibition. They form colonies in semi-solid media, and they have decreased requirements for growth factors. Some of these will become evident why they are so in a moment. But here on the right, your normal cells respect each other and stop growing when they touch. That's called contact inhibition. And the one on the bottom here, their cells are piling up. They're transformed. So chemicals can do this. Mutagens can do this. Viruses can cause transformation as well. But oncogenesis is separate. Oncogenesis is the development of a cancer. A tumor, for example, is swelling caused by abnormal growth of tissue. It can be benign or it could be malignant. And cancer is a genetic disease. 8 million deaths a year in developed countries. Why only developed? Because there are cancers everywhere, of course, but we don't have good ways of measuring them. You need about 12 mutations in different genes to cause a cancer. And these genes are involved in signaling pathways that govern the ability of the cell to proliferate, to live, to determine its cell fate, and to maintain genome integrity, which means correct mutations. So you need about a dozen mutations. So what happens typically is something will make cells continue to divide. Cells do not divide all the time. They're highly regulated. When they continue to divide, at each division, mutations get introduced into the DNA because the polymerase makes errors. And initially, if enough mutations occur in those 12 genes, then you get a cancer. But what makes the cells divide initially is a virus or a chemical or some other environmental cue. So these mutations can be inherited. So you can inherit 10 mutations from your parents, and then you just need 2 more to get a cancer. They can be caused by DNA damage, environmental carcinogens, infectious agents. They initially make the cells divide. They're transformed. And then sometimes those cells will get the right mutations to drive it to oncogenesis and form a cancer. So it's a really important distinction, okay? Transformation and oncogenesis are distinct. Transformed cells may not be oncogenic. It needs additional genetic changes. So this is an example of a transformed cell line that was injected into a mouse and formed a tumor. So after enough divisions, it had the mutations it needed to form a tumor. So we learned about this from studying viruses, what causes transformation and what causes tumors. And studying virus transformed cells gave us this insight into the events that establish oncogenic potential. No virus can do it all. Viruses don't actually cause cancer. They transform cells, and then the cells sustain mutations on their own, and they become cancerous or oncogenic. So, I mean, it's a fine line, right? Because without the virus, you would never get to that point. But as I said, viruses, carcinogens, genetics can all lead to transformation and cancer. So here are some human cancer viruses. Viruses are a contributing factor in about 20% of human cancers. We have both RNA and DNA viruses. We have HTLV1 and HIV1 and hepatitis C virus. Those are the RNA viruses. And these are the cancers they cause: leukemia, HIV, many cancers in different tissues and organs, and hepatitis C virus, of course, hepatocellular carcinoma. Then the DNA virus is Epstein-Barr virus, Burkitt's lymphoma, among other cancers, nasopharyngeal carcinoma, Kaposi's sarcoma, herpes virus, hepatitis B virus, human papillomavirus, cervical, penile, anogenital, head and neck cancers. Of course, that is what Henrietta Lacks had. She had gotten infected with HPV, and it caused the cervical tumor on her. We're not going to actually get to the DNA viruses today. We'll save that for another time. We're going to talk about RNA viruses that how they cause cancer, and then Merkel cell polyomavirus. So this starts with Dr. Peyton Rouse in 1909. He was working at the Rockefeller Medical Institute. That's what it was called back then. And a farmer brought him an English hen that had a tumor. It had a solid tumor called a sarcoma. So by now, in 1909, Rouse knows about viruses, right? We already know that they're filterable agents. So he grinds the tuber up, he filters it, he takes the filtrate, that's what passes through the filter. So if there are any viruses there, they pass through the filter. He injects it into another chicken, and the other chicken gets the same kind of tumor. So he says cancer can be caused by a viral infection. I would say cancer can be initiated by a viral infection because the virus transforms the cell, and then the cell does the rest itself. So what's going on here? Well, it took 50 years for people to believe Dr. Rouse, the longest incubation period for a Nobel Prize. He actually lived long enough to get it in 1966. And the virus he discovered, Rouse sarcoma virus, is a key player in 2 more Nobel Prizes. So Rouse didn't know what he was doing. But he stopped working on it himself for many years because he got frustrated and couldn't figure out what was going on. But figuring out what was going on started with Howard Temin. And in 1951, he went to Caltech, and he went to work with Renato Dulbecco. So there's Temin on the left there, and Renato Dulbecco here on the right. Dulbecco was a pioneer. He recognized when a technique was going to be powerful, and he had seen the plaque assays done for bacteriophages. He immediately transformed it changed the methods to be used for animal viruses. And then he thought, we can use cells to study cancer. So, Temin went to Caltech to work on fruit flies, but he switched fields and decided to work in Dulbecco's lab. And remember, up to now, the virus can only be studied in chickens. But Temin and Dulbecco said, can we do this in a culture dish? And he did. And so what he did, this is a description from the emperor of all maladies here. He added the virus to a layer of normal cells. The infection of the cells incited them to grow uncontrollably, forcing them to form tiny distorted heaps containing hundreds of cells that Temin called foci. The foci, Temin reasoned, represented cancer distilled into its essential elemental form, cells growing uncontrollably, unstoppably. So he wasn't quite right there, right? They're transformed cells caused by the virus infection. They're not yet cancers, but they're on their way. So here are 2 different kinds of foci that Temin observed when he infected chicken cells with Rouse sarcoma virus. You can see a focus here on the left and then a big focus here on the right. So these cells are piling up. They're pretty rare because not every infected cell is transformed. Okay, so what's going on here? How can a viral infection cause a cell to be transformed? Now, 3 things have to happen. You can't kill the cell, right? You cannot kill the cell. Because if you kill the cell, it's not going to be living forever, right? You have to reduce virus replication so the virus particles are not made. And then the cell has to continue to divide. It has to be immortal. So this is a special kind of a persistent infection. An infection that goes on the lifetime of the cell, but you don't make virus particles, and the virus is not killing the cell at all. So this was one of 3 pathways that were taken to understand the biology of cancer. So we have the study of retroviruses here, and we just learned about Temin in the 50s doing some in vitro studies. We have in vitro cancer biology, the cancer biologists trying to figure out where cancers come from, and then people working on DNA viruses. And together, these converge in the 60s and 70s to give us our present unified theory of cell growth control. And today we're going to just talk about retroviruses. And the story goes back to Rouse now. And so what you have to know is that there's another kind of retrovirus related to Rouse sarcoma virus called avian leukosis retroviruses, ALVs. These are endemic in all chicken flocks. You buy a chicken, you got ALV in it. It's not going to hurt you because it won't infect you. In 1908, Ellerman and Bang found that this virus causes leukemia in chickens. In 1908, they showed that before Rouse did, but people didn't think that leukemia was a cancer, so they didn't get much credit. So what happens is within months of hatching, most chickens in the world get infected with ALVs. You get leukemia in about 3% of birds as they age. The rest of the birds have a transient viremia, they become immune, and they don't get leukemia. But these birds, this 3%, go on to develop other cancers as they age, including sarcomas, the same kind of sarcoma that Rouse found. That happens in chickens that are first infected with ALV. So if you get virus from these tumors, you inject it back into a chicken, they cause sarcomas, not leukemias or leukosis, another word for leukemias. So Rouse isolated one of these viruses, Rouse sarcoma viruses, and many people subsequently did the same experiment and isolated slightly different viruses, and he was lucky. His was not defective. Most of the other viruses are defective that cause these tumors. I'll tell you what that means in a moment. So then the question began, how does Rouse sarcoma virus, but not avian leukosis virus, cause sarcomas? Remember, Temin has also already established that you can infect cells, chicken cells in culture and get foci, so you can start to study the basis for transformation. And so what Mike Bishop and Harold Varmus found is that Rouse sarcoma virus carries a cellular gene. It's an oncogene called VSARC. The cellular gene is called CSARC. A piece of the ALV genome is replaced with a segment of host DNA called an oncogene. So we have ALVs infecting chickens. A few of them get tumors, and those chickens with tumors, those viruses have picked up a piece of cellular DNA, which is an oncogene, and that's what's transforming the cell. And they got the Nobel Prize for this in 1989 for discovering this. And I interviewed both of them. We have interviewed both of them on TWIV. Well, actually, Harold Varmus was on TWIV 400, and then Mike Bishop I interviewed on my own as part of a series of interviews for the Virology textbook. So here's the story then. Most birds, many infected birds, 3% come down with a variety of tumors. The rare tumors have retroviruses derived from ALV. Most were defective. They were all different. Rouse was lucky. His isolate was not defective. So sometimes you have to be lucky, because you can only grow a defective virus with a helper virus, and he might not have known that. So when this experiment was repeated over and over again, the retrovirus genomes from each new solid tumor had different host DNA, not the SARC gene. They found all kinds of other oncogenes from the cell. A gold mine for molecular oncology. We can now identify the genes which, when mutated, cause cells to divide forever and to become eventually a cancer. And so here's a summary of a lot of that work. So here are what we call avian transducing retroviruses. And so this is the genome of avian leukosis virus, which infects every chicken in the world. It's a typical retrovirus. It has GAG, pol, and envelope genes. This is actually the mRNA of the virus. And then Rouse sarcoma virus picked up the SARC gene. It stuck it on at the end here. So it still has intact GAG, pol, and envelope genes, which are needed for virus reproduction. But the SARC is now added at the end. And this SARC gives it the ability to cause solid tumors, sarcomas. That's why the oncogene was called SARC. And then people isolated a variety of other kinds of tumors from these chickens and got different oncogenes. Myeloblastosis, a different kind of tumor. MIB oncogene. Another sarcoma here, erythroblastosis, reticuloendotheliosis. But look at these viruses. In them, the oncogene has inserted into a virus gene. So the MIB is right in the middle of envelope. And here it's in the middle of the polymerase. So that makes the virus defective. It can't grow on its own. So you need a helper virus, which would be avian leukosis virus. And people did the same thing with mouse tumor viruses. Here's murine leukemia virus, very much like ALV. It causes leukemias in mice. And mice infected with them come up with a variety of other cancers. And you can isolate from those cancers transducing retroviruses that have different oncogenes. Amazing. So they're called transducing because when they infect the cell, they're picking up a piece of cellular DNA and transferring it to another cell. So that's defective versus non-defective. Defective viruses need a helper virus. Right? The helper is ALV, and Rouse is non-defective, but AMV here is defective because the oncogene is inserted into the envelope gene. And these genes are deleted during oncogene capture. I'm not going to talk to you about how that happens, but when the retroviruses integrate in the host cell, a consequence is if they integrate near an oncogene, they can capture the oncogene when they leave the genome. So we have now found about over 60 cellular genes that are captured by these transducing retroviruses. We call them proto-oncogenes. In cells, in all cells, they control cell growth. Cell growth is highly regulated. Why? Because if cells keep dividing, they become cancers. So most cells in us are highly regulated in their division. And these normal cellular genes that are called oncogenes, they're called c-oncs, like c-sarc, c-myc, etc. And then when the retroviruses pick them up, they're called v-oncs, like v-sarc, v-myc. And sometimes, most of the time in the virus, there are other changes to these oncoproteins besides compared to what they were like in the cell, and that enhances their transformation capability. But all these oncoproteins encoded by oncogenes, they give the cell a green light up here. So this is a diagram of the cell cycle. And at the very top, we have mitosis when cells divide. Then we have a couple of phases. We have G1, where it's preparing for growth. S is the synthetic phase, where the DNA is being replicated. And then G2, it's preparing for mitosis. And now you have big cells that have grown. They have double the amount of DNA. They split in half. And that's mitosis. You now have two cells, and you keep going around that. These oncogenes give the cells a green light at this point. If the oncogene is present, the green light is on, and the cells keep going around this cycle. In fact, there's another checkpoint down here, this red bar, and we can talk about that another time. So there are two checkpoints to control the cell cycle, very important. Now, these oncoproteins are amazing. So here are the transducing retroviruses that have been isolated, and we have simians and birds and rodents here. And the protein picked up by each of those is involved. Each of them is a different part of this mitogenic control pathway of the cell, the pathway in the cell that controls whether the cell is going to divide or not. And it consists of growth factors and growth factor receptors. And when the growth factor binds a growth factor receptor, it starts a series of protein phosphorylations, which end up in some proteins, transcriptional regulators, going in the nucleus and turning on genes, which in turn encode proteins that make the cell divide, like DNA polymerase and other proteins as well. And so if you turn on any of these, the synthesis of any of these proteins, you get uncontrolled cell growth, and that's what these retroviruses do. You can see some of them encode the receptor protein tyrosine kinases. Some of them encode other intermediates in this pathway, and some of them even encode transcriptional regulators that turn on other genes. So the retrovirus deposits these oncoproteins in the cell. The cell keeps dividing. It's transformed. It's immortal, and eventually it will sustain the number of mutations needed to cause a cancer. So the mitogenic signals that are controlling the cell cycle division were revealed by studying transforming retroviruses. We call these dominant oncogenes because you put one copy of the gene in a cell and it makes the cell divide forever. Really amazing. And finally, let me just tell you that there are three ways that retroviruses transform cells. So there's rapid tumor formation, like Rous sarcoma virus. It takes about 2 weeks in the chicken to get a tumor. And that's because the virus has an oncogene in its genome, and the protein is made immediately when the virus replicates. We have some viruses with intermediate kinetics, like avian leukosis virus. It takes months for the chickens to develop leukemia after they get infected. There's no oncogene in ALV. But what happens is it integrates next to an oncogene and turns it on. I'll show you a picture of that in a moment. And finally, we have some viruses like human T lymphoma T lymphotropic virus, where it takes years to make a tumor. There's no oncogene. It doesn't integrate next to an oncogene, but the virus makes a regulatory protein that activates cellular proteins that makes the cells divide forever. And that's typically IL-2, as you'll see. So here are the three genomes. Transducing retroviruses, rapid transformation, 2 weeks, they carry an oncogene. Intermediate transformation, months, the retrovirus doesn't have an oncogene. It integrates next to one, and it turns it on because the LTR has a promoter. And finally, the transactivating retroviruses that take years to cause a tumor or transform cells, they make a protein called X, which is needed to activate the promoter in the LTR of the viral genome. And that X will also activate cellular genes, in particular the IL-2 or the IL-2 receptor gene. And when you do that, that makes T cells divide forever, and that gives you the lymphoma. So that's how these retroviruses cause cancers or lead to cancers. Now, it's really important to understand that retroviruses transform cells as a mistake. It's a byproduct of their reproduction cycle. Their DNA has to integrate into the host cell chromosome, and then sometimes it integrates next to an oncogene, and then you have troubles. There's no obvious requirement for transformation or oncogenesis for the reproduction of retroviruses, with one exception, the walleye dermal sarcoma virus. This is a tumor of walleyes, and it's caused by a walleye dermal sarcoma virus. And the tumor formation is absolutely essential for propagation of this virus. It's not for the others. It's not at all. As if I didn't have enough problems, and my soundboard just shuts off. My God. So the transformation of the walleye cells is important for spread of this virus, but for the other retroviruses, transformation and tumor formation is not essential. Okay, so that is our little mini lecture on how RNA tumor viruses transform cells. I think this is just a fascinating story, right? And we learned so much for that. From that. I love that story. And we'll talk about the DNA tumor viruses another time, perhaps next week. Now back to you, folks. I need to wear pink glasses. I don't have any pink ones. I have blue, I have clear, I have multicolor, but not pink. Okay. Let's see. Let's get down to. I texted Les and I said, "Tell people to wait." And he said, "It's okay. They're very chatty. Your audience is very chatty." It's been Curly Dog asks. I mean, this is a topic Daniel and I talk about all the time. It's been more than 6 months since my last poke, so I'm presuming this is a SARS-CoV-2. Yeah. Is it really time for another SARS-CoV-2 vaccination? Here's my take. The CDC is saying if you got the fall modified vaccine, right, the Omicron-derived virus, now you should get another one if you're 65 and up or have some health problems. So this is because if you're at that age and you have other health problems, when you get infected, after three months after you got the vaccine, your serum antibodies and T cells have declined to low levels. That's the way it works. And then when you get infected, you have to wait for those memory cells to get reactivated. And older people with comorbidities can't handle that. They get very sick, even though eventually the immune response kicks in. So the CDC is saying, let's get a booster every 3 months, which I don't think scientifically. I'm not giving you medical advice. I'm telling you the science, and you can decide what to do. Doesn't make sense. What I did is I took the booster last fall, and now I'm just going to, if I get a respiratory infection I'll see if it's COVID. If it is, I'll take Paxlovid. Now, I understand Paxlovid is not available in every country, but the alternative is to take a booster every 3 months, which doesn't make any sense. We don't do that with any vaccine. Even flu, we do once a year. So that's what I'm saying. So I don't know your health situation, right? If you're healthy, even if you're 65 and over, just take Paxlovid if you can, if you test positive. Because I don't think it makes sense to keep getting boosted every 3 months until they figure out how to make a broadly protective vaccine, which is a goal, and it should be a goal, right? You shouldn't have to get a vaccine so frequently. Here, Curly Dog. Chris Lipowski in Canada near Montreal with a view of Vermont. Nice to have you here. And Markle is in the east end of Long Island. I don't know if I got all these individuals. Yeah. We're all up and running now. It's really good. Tom is in Oregon. What? Internet down somewhere? Yeah, it happens. But it came back up. We got it. We first did what we're doing now is a hotspot because I don't want to change it midstream, but the internet's back up again. Rona is on Long Island. We've been in the Utah desert. Karen is from Maine. This is before I launched. Sunset Bay, Asheville, North Carolina. Claire's from the UK. This could be a short office hours. No, it's not. It's not a short one. We did it. We I don't want to just leave you guys flat, but also I prepared and I wanted to do it. Bad Cat is from Central Maine. Mr. Doreen is from Elgin, Illinois. Is anyone from, I don't know what that flag is, UAE? Not sure. Lebanon? I don't know. Carol is from California, our favorite nephrologist. I don't go home anymore to do these. I do them here at the incubator because it's too hard to get home in time to do this by 8 o'clock. Oh, Jolie is in the UAE. That's right. Charleston, South Carolina. I'm getting to the questions. Pete has got his cup of tea and is in London. Laurie and Mike are in Seattle. Frank is in Santa Barbara. Okay, Kang. What triggers a cytokine storm? Is it a positive feedback loop where a cytokine continues to stimulate a more intense reaction? Does an increase in cytokines make a mild cold severe? So cytokines are produced when an infection is detected. So you get some virus coming into your upper tract. It's sensed by the innate immune system, and then cytokines and chemokines are produced. And that leads to inflammation, which is swelling, redness, heat, and pain, because immune cells are coming in, so you get this swelling, and then fluids are coming in from the tissues. So that's inflammation, and inflammation is regulated because it can be dangerous, right? Redness, swelling, heat, and pain is not good to have all the time. So there are regulators of chemokines, and after the inflammation has gone to a certain point, the anti-cytokines are made and they shut it down. And so that's the normal progression of things. So a cytokine storm can happen if these regulators aren't functioning properly. They're not made at enough levels. But also we think if an organism reproduces to high levels, if the innate immune response doesn't control it, the virus can reproduce to high levels, and then you get an overstimulation of cytokine response, because now you have a lot of virus, so you make a lot of cytokines, and then that crosses a threshold where it becomes pathological. So I think it involves a number of components like that. And yeah, you could have a mild cold, and if you have severe inflammation, it could make it worse, for sure. So, most SARS-CoV-2 infections are rather mild or moderately mild, but a small number, about 20% or so, end up with severe lung inflammation. Not so much virus reproduction, but lung inflammation. And you can't breathe, you have low oxygen saturation, you have to be intubated and given oxygen. So that's a cytokine response. It's an inflammatory response. Yep. A 96-year-old has also been offered a jab. Yeah, I mean, if you can't get Paxlovid, you can get a jab, but I don't think every 3 months a jab is sound public health policy. You know, I have to tell everyone, last Friday's TWIV, 1105, talked about an experiment where they made mice lacking antibodies. And when they infect them and then challenge or vaccinate and challenge with SARS-CoV-2, the mice are fine. They prevent any disease whatsoever, and they clear the infection. So you don't need antibodies to do that. You just need T cells. But those are young mice. So I think in young people, that explains why you still get mild disease even when variants are emerging, mild to moderate. But in older people, your T cells aren't good enough because from roughly 65 years of age and up, when you get an infection, a new infection, it's hard for you to make good new T cells. You can make a memory response to infections you've had younger, that's fine. But SARS-CoV-2, a new virus, a lot of older people can't make good T cells, and so they have a problem clearing the infection. So what do we do about that? We have to figure out a vaccine that bypasses this problem, and it's not a trivial issue. So that's the story. Now, one more line from that story, which is really interesting. What's going to happen over the years with SARS-CoV-2? Well, I think we have a clue from the common cold coronaviruses. There are 4 common cold coronaviruses that circulate globally. They infect everyone every couple of years, and most of the infections are quite mild. The reason is those viruses came from some animal into people many years ago, hundreds of years ago. And there was probably a pandemic where elderly people died in high numbers. But eventually, the elderly population moved on, and then it was only people constituting the population who had seen the common cold coronavirus at a young age, and they were able to make good T cells. And so as they age into their 60s and 70s, they can have a mild infection with a common cold corona because they have memory T cells that are good. So that's where we have to get with SARS-CoV-2. And it may take 50 years for the elderly population to depart, but then they'll be replaced by a population that saw the virus the first time at a young age, and that's the key to making it mild. It's not the virus changing. It's population immunity in relationship to age. Okay, that's what I think is going to happen. Okay, so Lise wants to know how to get reservations at Isodi. I have no idea. I never tried. Go somewhere else. There are plenty of other good restaurants. Went to a good one for the textbook. I can find it out for you. It's really good. You can get a reservation. It was so hilarious to see one of the big disinformation screwballs brag that he had challenged you and Dr. Offutt to a debate. He is a compulsive liar. Did you see what did Alan Dove call Twitter X? Zitter? Anyway, so the infamous person in Rutgers, New Jersey, put out a list of people who are, what's the word? Let me find it. Hang on. Here, deceivers. We're deceivers. He put out a tweet, a multi-thread tweet. Here's the attempting to deceive category. It includes Xi Zhongli, Lin Fo Wang, Peter Daszak, Peter Hotez, Anthony Fauci, Francis Collins, Christian Anderson, Bob Gary, Eddie Holmes, Andrew Rambo, Angela Rasmussen, Saskia Popescu, Linda Safe, and Vincent Racaniello. We are all deceivers. And then he goes on to name others. We're deceivers because we don't buy into his crap that SARS-CoV-2 came from a lab, which is, it's all crap. There's no narrative. There's no data. And, you know, all the good data says it came from nature, but he says that's a fraud. That's a fraud. Don't you know those papers are all fraudulent? Really? Anyway, so I'm a, what am I again? I'm a deceiver. The big deceiver. Walter is from North Central Massachusetts. And Steph, thank you for mentioning the tethering. I am the most uninformed techie on the planet. Right. I'm looking for questions, folks. That's why I'm just being a little slow here. We're just seeing everyone happy when I came. If the virus contaminates and induces infection, maybe must avoid zinc supplements. I don't know what this question means. Not all viruses require so much zinc. They can require trace amounts of zinc for enzyme function and so forth. So are you saying that if you take zinc supplements, it may aid and abet virus reproduction? I don't know if there's any evidence for that. I think we have plenty of zinc in us already, and anything you take is an excess unless you have some condition, right? Pete says, 7 years today, I said goodbye to my last dyno fossil car, electric car ever since. Well, that's cool. Lucky you. I just don't have any car whatsoever. I'd like to try and do that. Is there a concise source of info about the freshwater parasites in Southeast Asian rivers? I think that's very granular. The best you're going to do is parasitology book, which you can find for free at parasiteswithoutborder.com. And you can look in that, but it's not going to be concise, right? Because it's a textbook. But I don't know of anything else. Hunter says, check for viruses. Yeah. Check for viruses. Let's see how my hotspot, so one of these hotspots is running on battery. So let's try and see if I can now switch that to the incubator. Let's see if it works. Yeah, it works. Yep, it was working. I did my stream computer switch and it's working. But I'm not going to do the main one here because it'll burp and maybe Ecamm won't like that. Siddhartha, thank you for your support of science communication. We appreciate your support. I met Siddhartha a year ago at TWIV 1000. Thank you very much for coming to that. That was pretty cool. Internet on phone is often more reliable than landline internet, strangely. Is that right? So if you have a good cell signal, it's reliable? Because as you know, you can move through places and your phone signal dies, right? So I'm on a train and we go through this portion of the ride when there's no bloody cell signal. But I guess if you're sitting here, and what do we have here? We have 5 bars of 5G. It's pretty good. 5 bars. Yes, the internet is back on. That's interesting. Merchlin Vin from the Philippines. Hello. Can't pronounce your name. But hello. Portia says hello. and somewhere in California, I believe, is Portia. Why does hyperplasia become cancer? So hyperplasia is simply uncontrolled division of cells, right? And you can pick that up on a pap smear, for example, hyperplastic tissues, and then it gets enough mutations. It gets enough mutations to now become a cancer. So in 12 or so genes, right, that are important, not just for cell growth control, but also the ability to spread to other tissues is part of these changes caused by these mutations. So that's the difference. The cells divide and divide, sustain mutations until they get the right 12 to make it a tumor. And why does it happen? Because DNA replication is error prone, and cells are not meant to divide forever. They are meant to divide intermittently. And then the ones that do divide forever, they slough off. So your intestinal epithelial lining divides continuously, and then they come off after so many days. So they don't normally have the ability to become cancers, but they do. They can in some cases, as you know, of course. By genetic, you don't mean I do mean heritable. Yes, you can inherit mutations in some of these hallmark genes, these dozen or so genes, and from your parents. So you could inherit none, in which case you'd have to live a long time to sustain the other mutations. You could inherit 6 and have less time. But the point is that we're always assaulted environmentally, right? UV light, chemicals, air pollution, all of this is assaulting us continuously. And that contributes to it. So, ideally you live on a pristine tropical island with nothing but the pure air. And be careful what you eat, and then maybe you don't get cancer. But you can't. You can't do that. So heritable is all that it's about. Yeah, necessary but not sufficient. Yeah, transformation is necessary but not sufficient for oncogenesis. That's a good thing. The other RSV, yes, we have Rous sarcoma virus and respiratory syncytial virus, which it should be abbreviated RS virus, just RS, because you don't confuse it with RSV, but people just don't do that. What is the Jeopardy question? What's the Jeopardy question, Tono? Add it to if you're still here, add it, please. I want to know. Lori recognizes Dulbecco from tissue culture media. Yeah. Dulbecco's MEM. Minimal essential medium. Yeah, so he did all kinds of things. It's really good. Your interview with Bishop was great. Thank you. I had a good time interviewing those two individuals. Yep. David Baltimore and Harold Varmus are guests on episode 904 of TWIV, 50 Years of Reverse Transcriptase. Yeah. I wish we could have had Harold Varmus. No, we had Harold Varmus. Howard Temin on that episode. But, of course, he passed away a number of years ago. Tona says, how many years away are we to know why 40% of women with BRCA don't get breast cancer? So that's all about genetic penetrance, right? Not every mutation, not every genetic change has a phenotype. And I think part of it is that you need other changes as well, most likely, to cause cancers. But I don't know how long it'll take. I think I can't put a date on it. I just don't know. I don't know if this is related, Markle says, to the chicken tumors, but sometimes the wild turkeys who nest in my yard will have poults who, as they grow, get a type of growth which grows in the GI and out of the mouth. I don't know. I don't know anything about chickens and turkeys. It could be a tumor, And if it is, it might be a viral tumor. I just don't know anything about it. Visto says, could Tasmanian devil infectious tumor, the Tassie devil, have a viral origin? I don't believe it does. That's a tumor that's spread by cells. The cancer cells go from one animal to another, and they establish them. So instead of a virus infecting a cell, going from one animal to another and causing the cell to be transformed and eventually leading to a tumor, it's the actual cancer cells of the Tasmanian facial tumor that get transferred. These animals fight and they transfer their cells. And they're so genetically related, these Tasmanian devils, that the recipient doesn't reject the donor cells. How about that, right? So there's no evidence that a virus is involved in that. That's there are 80% of human cancers are not of viral origin because there are many other insults that can make the changes that lead to transformation and then oncogenesis. Andrew says, I can remember where I was when I learned that viruses can cause cancer. I was listening to a podcast on HPV, walking on a dike by the Weser River in Bremen. Oh, interesting. So human papillomavirus, a big player in that was Harold Zurhausen, who got the Nobel Prize for that. He passed away recently, but I interviewed him in Manchester a number of years ago. You can find that. I think you can find it. It's on YouTube. Just search for Racaniello and Zurhausen, and you'll find it. I'm going to do that right now. Look, YouTube. We're going to go Racaniello, Zurhausen, and there it is. Harold Zurhausen, Human Papillomavirus, episode 80. Let's Microbe World. It was a different podcast, but let me paste this into the the stream so you can watch. Harold Zurhausen, really good. Tryon wants to know what viruses can induce oncolysis, lysing tumors. So this is a very nuanced question. So some viruses can lyse tumors, like Retrovirus and measles virus and adenoviruses and Seneca Valley virus. But that is not the only way that, and even certain polioviruses are being investigated for treating glioma. But that's not the only way that the tumor is cleared, right? Because you never get an infection of all the cells in a tumor. What happens is the lysis of the tumor releases tumor antigens, which then the immune system, it kind of wakes up the immune system because the immune system is ignoring a tumor, right? That's why it's there. And so the release of these antigens stimulates inflammation, or the cytokines and chemokines, and then immune cells come in and destroy the tumor. So it's a synergistic effect of the viral lysis and the immune system being alerted to, hey, there's something going on here. Yeah. Now people are asking for Amy. Amy is currently in Switzerland. So it is 9, 10, 11, 12, 1, 2, 3 a.m. So not likely that she is here because she's awake. She's not awake. Yeah. And, the lost audio was like a minute. I have this board here with 12 inputs. It's just cut out. Like, what is that about? I have to turn it off and on again, which is not a big deal, but why does it do that? Is that like a glitch in the machine that every now and then some electron goes awry and it turns it off? Why? Why do things happen? I can't explain viruses, but these other phenomena, electrical phenomena, I can't get it. I don't get it. Thank you, Bree. Bree says, thanks for always being gracious and giving us your time and knowledge. A true teacher. This is what I want to do, is teach you folks. I love teaching you folks. And It's my pleasure to do this. And yes, the scientists who put their hearts and minds into the research that I summarized very quickly, a lot of people do that. They do it because they're curious and it helps humanity. It's really great. Science is great, and people should not poo-poo science. It's not fair. Oh, Visto, do the walleye fish cells shed and infect other fish? Yes, that's what happens. The cells fall, the tumors fall off the fish, and they break open in the water and release virus, and that infects other fish. That's how I understand it happens, right? Retroviruses are very similar to host retrotransposons. That's right. So host retrotransposons are genes that move around the genome by reverse transcription. So retrotransposon, reverse transcription. And we think those are the ancestors of retroviruses because they have a gag gene and a pol gene, but no envelope. So they can't make particles. They have two LTRs and they have the reverse transcriptase. And so at some point, one of these captured an envelope gene, and off it was to the races. It became a virus, a retrovirus. Yes, it's really good. So yes, the mini lecture really helps solidify things for me after the regular lecture. It is just like attending real office hours. So yes, this lecture I only gave a few weeks, maybe last week, right? So those of you who have seen it have heard it, but it's an abbreviated version, so you can focus on a few points. It's short, and of course, I say slightly different things in it. So I think it's useful to emphasize it, as John says here. Yeah. So prior to the mid-20th century, what was leukemia thought to be? I don't know what people thought it was. It's just like, infectious diseases were thought to be bad air. You're under the influenza of bad air. We didn't know. And people would think, well, if we put leeches on people and bleed them, we can cure them. Leukemia, I don't know the history of that, but maybe in Emperor of All Maladies, you can find that out. Hunter had the pleasure of meeting Renato Del Becco at the Salk Institute when my mother worked there as an administrative assistant back in '79. Yeah, that's cool to meet some of these famous people who did cool things. John says, I know it isn't Amy live from New York, but her appearances on Twivs are really good to revisit. Yeah, Amy used to be up in the lab at Columbia working. I would be at home and we'd do a stream. She would do her experiments, do the stream from like 8 to 9, and then go home and eat dinner. Yeah. Midge says, listening to your old TWIP, so interesting about parasites. And Dixon blows my mind what he knows about parasites as you do with viruses. Yes. If you work long enough on something, you may get to understand it somewhat. Not always, because not everybody learns as much. Patricia wants to know why HIV vaccine is so impossible. Okay. So vaccines don't prevent infection, right? Our current technology doesn't allow, and I don't think our immune system allows it. Why? Well, let's say you've never been infected with HIV and you get infected. The virus comes in, it starts to replicate. Then the immune system kicks in a couple of days, the immune system ramps up. But within weeks, the virus has already integrated its genome into long-lived lymphocytes, and there's nothing an immune response can do to prevent that. And so you still get AIDS even though you're vaccinated. Many vaccines have been tested. They cannot respond quickly enough to prevent the establishment of that latent reservoir. It takes about 2 weeks to establish after infection. So you would have to have a sterilizing vaccine in order to prevent HIV/AIDS. Yeah, it's a really tough ask. How does fasting change cell growth? I don't know the details. Amy would tell you exactly what's going on here because there are regulators in the cell that control cell growth, that respond to nutrient levels. In part, so cells divide when growth factors bind to cell surface receptors, like I showed you in the mini lecture. They bind if growth factor is present, which you would get from having a meal, for example. It would bind the receptor and it would turn the cell division cycles on, and so the cells would divide, and that would make the tissues bigger. So that's the extent of my knowledge, but there are other controls as well. So Wondering World says, I'm pro-vax, but with the amount of COVID around now, I think our immune systems are going to be boosted naturally anyway, more than every 3 months. It could be. But remember, yes, so if that's true, I don't know if it's going to be every 3 months for everybody, right? You want everyone to be protected from severe disease. But let's say if it were for everyone and everyone got boosted every 3 months, that would be a natural solution. Then you would have high antibody levels as a consequence. But I don't think we can depend on everyone being boosted synchronously, right? In time for the season, for example. So I think it works for some and not others. Visto says he doesn't like retroviruses. They are like home invaders. They break in and trash your DNA. How rude. Yeah, but they're not doing it on purpose. They have no intent, right? It's just they're parasites. They're just happening. What do we have here? We have 200 people tonight. I'm very impressed because it's just me. So thank you all for coming for Vinny. And 132 likes. So we could do better on the like button. So please hit the like button. It's just below your video screen. And why do I want you to do that? We don't make money. It's just because other people may see it as they're surfing YouTube and come over and learn something. You know what Socrates said? I cannot teach people anything. I can only show them how to think. I think that's good. And it's what we try and do here on Microbe TV podcast. We try and teach people how to think, or not teach, but show people how to think, because we can't teach you anything, right? I like that. Abdur, thank you for your contribution to science communication. Assuming this is related to oncogenicity of HBV and HCV, interesting how RSV is ubiquitous in avian species, kind of the BK and JC in humans. Okay, so Abdur Rahman is referring to two viruses, BK and JC, polyomaviruses, that pretty much infect everyone without causing any problems whatsoever. So they're ubiquitous in that sense. But the ALVs, of course, see, the difference is the ALVs integrate. And so at about 3% of the time, that integration is next to an oncogene, and that causes the transformation of the cell. Whereas the BK and JCs never integrate. However, for the DNA viruses, you don't need the whole genome to integrate to transform cells. So you'll see next time when we talk about DNA tumor viruses, you just need the oncogene of the virus to integrate and be produced continuously. So I'm thinking now, JC and BK are small DNA viruses. They definitely need to turn on cell division in the host because they use the DNA polymerase of the host. So does that happen? And if so, if it does that, they must kill cells and never lose their lytic nature. Otherwise, they would cause tumors. They don't. That's very interesting to think about. Thank you for jogging me. HBV and Hep C, there don't seem to be oncogenes involved in their cancers, but rather continuous destruction of liver cells and regeneration and destruction and regeneration. So the liver cells are dividing continuously and being destroyed. So that eventually leads to cancer because, again, you're dividing all the time. How do you explain bovine leukosis virus causing tumors in only about 3% of cases? I don't have an answer. It's a very interesting question. Bovine leukosis virus is interesting because it has an oncogene, which is an siRNA, that silences something in the cell that makes it become transformed. And I don't know why it's only 3%. Yeah, I mean, it could be that they hit the 12 mutations, but I don't think that, well, maybe it's like ALV. Maybe it has to integrate. So I don't know. I'm just speculating out loud. So if it were ALV analogy, the BLV has to integrate next to an oncogene, but I don't think that's the mechanism. Bovine leukemia virus cancers. We're not going to get it quickly. Yeah, 2 to 3% of cattle develop cancer and die. The incubation period is relatively long. Huh. So I do think that this is the one with the siRNA that is the oncogene. But I don't know how that would relate to rare formation of tumors. It sounds like ALV in terms of its rarity. I'm going to look that up. That's a very interesting question. Let me see here. Let me write it down. Bovine leukemia virus mechanism of oncogenesis. Why 3%? Could be an exam question, right? Oh boy, I went all the way to the top of the comments now. Where are we? TWIP, I got that. Rahman, yeah, here we are. Back to the future. John says he's in the free dumb state, Florida. Mice lie. Yeah, well, they can. Yeah, they do lie. So I don't know what the mouse not having antibodies tells us about human infections, but, plus or minus antibody is a pretty straightforward result. But, yeah, I agree. Something else could be going on. We talked about that on the TWIV. Well, it's a situation with H5N1 now in cows. How concerned are you? Okay, so cows have been infected. Goats have been infected in several states. And this is because it's now in many, many birds globally. Usually it was a seasonal infection in birds, and now it's crossing seasons. And so many terrestrial mammals and water mammals actually also are being infected. So there's a lot of virus, H5N1 virus around. That's why these other mammals are getting infected. And then a human got infected in Texas. So how concerned am I? I think it's always concerning when viruses are in more hosts. These H5N1 viruses have been circulating for 50 years, and they have not acquired the ability to transmit effectively among humans. That doesn't mean it will never happen, but I think it tempers my concern a bit. I asked a couple of influenza virologists last year at a conference, are you worried about H5N1? And Ron Fouché, who works on it, said he's not worried about people. He's worried about the animals out there, like the sea lions that got wiped out off of Peru and so forth. So that's his take as an expert. There hasn't been an increased number of human infections coinciding with this increased endemicity of the virus in birds. But if it can infect different mammals, then there's the opportunity for the virus to recombine with influenza viruses in those mammals and produce something different. So I think we should be ready. Let's put it that way. I'm not going to lose sleep over it. I lose sleep over other things. But I'm not losing sleep over H5N1 because we do have antivirals. We have Tamiflu, Relenza, and Baloxavir, which are effective against that virus. We can make vaccines. People are testing mRNA vaccines right now. So I think we would be ready in the event of a pandemic caused by H5N1. Now people say, well, won't it be highly lethal? Because in humans there have been 800-ish infections and about 50% mortality. That's really high, right? But I don't think it will be that way once it adapts to people. I think it's that high because it comes into a new host and the host goes crazy. There's a lot of inflammatory cytokine storm type things, right? So I think if it does acquire transmissibility, it's likely to be of vastly reduced virulence. And also I think the 50% is an overestimation because it's the really sick people who go to the hospital with H5N1 and they die. And we don't know if there are lots of mild infections out there because they don't go to the hospital. Some surveys have been done in countries with large poultry industries, and they suggest that there are a lot more people infected than we know of, and that there are mild infections. So, they get a cold and they don't do anything about it. So that all those things temper my worry, but I think we should be paying attention for sure. Okay. Can the body's production of T cells be exhausted by repeated vaccination every 3 months? I think anytime you activate T cells, you have the potential for exhaustion. But the guy in Germany got 217 doses of mRNA vaccine, and he doesn't have any exhausted T cells as far as we can tell. So that's just one person, and everybody's different. So I would say it could, but I don't think it's a common observation. And we're going to see, because now there are people that have 4 and 5 and more boosters, so vaccinations, COVID vaccinations. So we're going to see. So I would say it's possible, and we'll see, but maybe not likely in most people. Is there any research about using viruses for insect pest control? Absolutely. There are many insect viruses, and many of them are being tested. The one that comes to mind is cricket paralysis virus. Yeah, it's a picorna virus like polio that infects crickets and it paralyzes them. And so it's being tested for pest control in some places. And there are many other insect viruses as well that are being investigated for sure. Yeah. And the thing is, the insect viruses are harmless to us as far as we know. When you eat cabbage, you eat a lot of insect viruses because the insects that have the virus are crawling on the cabbage, in the fields, and there's no way to wash it off. But it's harmless. It passes right through you. What are the key mechanisms by which SARS-CoV-2 can invade the central nervous system? And what are the potential long-term neurological consequences? Well, I think the most likely route is through the olfactory epithelium. So somewhere up here where your nose brings air in, we have the olfactory sensors so you can smell. And those are receptors on the mucosa, and they're wired into the brain. Right? So the SARS-CoV-2 replicates in the sustentacular cells, which are support cells in your nasal epithelium that are important for the function of the neurons that sense smell. And that's why you lose your smell. It's not because it reproduces in neurons. So some people have suggested that the virus can get in that way. there's not much of a viremia, so I doubt that the virus is getting through that way. Now, measles virus can, in rare cases, get into the CNS. 1 in 1,000 measles cases, you can get encephalitis. And it could be that there's a route from the lung to the brain for that. And then enterovirus D68, which is a respiratory infection with no viremia, it gets into the CNS. And maybe it's by nerves. We don't know. But anyway, so those are ways that SARS-CoV-2 might get in. The paper we recently did on twin, which hasn't been published, the podcast hasn't been released yet, shows that if you make brain organoids or if you make brain slices from people in culture and you try and infect them with SARS-CoV-2, the infection is very inefficient. Very few cells are infected, and they make very few infectious particles. So my opinion is, based on that the brain is not going to be crazy infected with SARS-CoV-2 virus particles. It's not going to be crazy infected. I think there may be, foci of infection here and there. What they do, I don't know. I think that neuroinflammation is possibly a more serious consequence. So cytokines can be produced locally in your lung, but they'll diffuse and they can get into your brain and cause these neurological effects. So what are the long-term consequences? Hard to know about long-term, right? So I think long COVID is instructing us that a lot of people have brain fog, cognitive issues, even motor problems. How long they last, we don't know. But they're going to be, we talked a little bit about this on the twin this week in neuroscience. They're going to be a consequence of probably the neuroinflammation. And so it's possible that they're transient, or possible in some cases that it's permanent. Is there any other benefit of the COVID booster shots that have been investigated? Well, 3 doses in general is good to reduce the incidence of long COVID, right? As well as reducing the incidence of death and progression to hospitalization and so forth. So that's the only one I know of that it is beneficial not only for preventing disease, but also reducing the incidence of long COVID. I like that list. Flattering to be with Francis Collins. Yes, Alan Dove was annoyed that he wasn't on it. The deceivers are a distinguished bunch. I would be delighted to be included. So there are really good scientists on that deceivers list, right? So what he is saying is that these are all bad scientists because we don't agree with him. I don't know what his issue is. I don't get it because there's just no coherent story for a lab origin. All the coherence is in natural origin. So I don't get it. I have not had a COVID shot for 2 years. I wear a mask into crowds and shopping centers. I think a mask is very good, and that's one way to prevent getting infected. Yeah. Write a checklist for what to do next time it happens. You have to write things down like this because this is a rare event. And you don't remember things that you do infrequently, right? So I do this PowerPoint slide thing with the stream once a month. That's not enough for something to be burned into your neurons. Once a month is not. So, writing it down. The question is, Andrew, where to put this list? These stickies are not going to cut it. They're all over my desk now. I don't like it. It's a mess. I have sheets of paper here, just sheaves of paper. I don't like it. I have the little field notes. I wrote in those. I filled it up, but I'd stopped using it. I don't know why. I used the notepad on the computer. Nothing really resonates with me. Nothing resonates except my brain, and it's fallible, right? John wants to know, why is it so challenging to make mouse models without antibodies? So you can easily make mice that don't have B cells. Those are pretty easy to make. But then you're not just missing antibodies, you're missing the whole cell, and that cell could be doing other things besides making antibodies, and it changes the architecture of the lymph node completely by not having B cells. So that's a problem. And then with T cells, you can remove T cells, but then when you add them back, you don't know if they're going to the right places and so forth. So that's why it's good to keep the cells and remove their functionality. So that paper, they made a mutation that removes the ability of B cells to make antibodies, but there's still B cells there. It's really, really brilliant, I think. It's very cool. What evidence supports or undermines the lab leak hypothesis compared to natural spillover? We've talked about this for 2 years now, Spartacus. There's no evidence for a lab origin. It is all speculation. Yeah, they were working on SARS-CoV-2 at the Wuhan Institute of Virology. No, they weren't. They were working on other coronaviruses that are not close enough to be SARS-CoV-2. So what's the evidence? Oh, the Chinese don't tell you the truth. Not evidence, not scientific evidence. This is all hearsay. People are committing fraud. You know, this first paper on the origins, the proximal origins of SARS-CoV-2, it came out in the first year. This guy who called me a deceiver says it's fraud. It's an opinion piece. There's no fraud there. It's nonsense. Oh, they said the fear and cleavage site is out of place, doesn't belong. If it were so out of place, it would be gone from the tens of millions of genomes that have been sequenced. It's there and it hasn't changed an iota. That tells me it's been naturally selected. So there's a lot of evidence for natural origin. First of all, every other coronavirus has come from animals. So let's not ignore precedent, folks. Do you not give that any credence? Apparently not. Every human virus has originated from an animal. There has been no lab leak leading to a pandemic. And don't tell me 1977, I'm not saying to you, Spartacus, don't tell me 1977 H1N1 pandemic. It was deliberately released as a vaccine trial. And don't tell me the Sverdlov experiments in bioweaponry, they were deliberately making a weapon. Didn't get out. Okay, so there are no epidemics caused by lab leaks. And there's plenty of evidence that all human viruses come from animals. And in this case, the epicenter of the early infection, the earliest cases were around the Huanan seafood market, which sells mammals, including raccoon dogs and civet cats. And there are 2 lineages of virus that originated there, and probably more, but we've detected 2. So people say, oh, someone could have been working in the Wuhan Institute of Virology and contaminated themselves and then went to the market. No, that's not how it works, bub. If they did it at the Wuhan Institute of Virology, that would have been the epicenter. It's epidemiology 101. I'm the biggest critic of epidemiology, but that makes sense even to me, okay? So no evidence. There's no coherent story for a lab leak. In fact, they say, oh, it's either a lab leak or a deliberately made virus. Well, we'll take your poison, pick it, okay? And then try and defend it. You can't. There's no coherence. People have an agenda. They're stupid. They want attention. I don't know what it is. If I say it's natural origin, I don't get any attention. That's fine. But if they say it's a lab origin, they get a lot of attention. And I think they thrive on that. So that's the story. Have I met Dr. Mukherjee? No, I was on a Zoom call once. I haven't met him, but he's an oncologist at Columbia. Yeah, he's my colleague, right? The Deceivers reminds me of the famous Irascibles group of Abex artists from the 1950s. You should get together for a group photo. It's now famous in the Abex world. That would be fun, right? Get all the Deceivers together and make a big picture, maybe on the steps of a brownstone in Harlem. Anybody get that reference? Probably Portia would. Southern California, Santa Cruz. There you go. Thank you very much. How about the CPE rate? When we inoculate the SARS-CoV-2 in cell lines from swab, we observe the CPE to be fast. It's slowed down. I don't know what you're asking. I don't know what CPE rate doesn't mean anything. Did you see Akiko made the Time magazine most influential people? Okay, so in my opinion, influenza can be good or bad, right? Hitler was on the cover of Time, wasn't he? As being influential in a bad way. So I think she's done some good things, and I think a lot of her stuff is hyperbole and overblown, frankly. And to say long COVID is 15% of all infections is just wrong. Anyway, congratulations, Dr. Iwasaki. If one is repeatedly injured at a given site, e.g. lung inflammation causing repeated cellular repair, can the higher rate of mitosis eventually result in cancer? So that's what happens in HIV-induced cancers. The virus is continually replicating. The immune system is continually stimulated. Cytokines are made, which are mitogenic. They cause cells to divide. And so you get various tumors as a consequence of this incessant division. And that's what happens with the liver cancers caused by HBV and HCV. It's a case of a lot of cell division. So yes, that can be a cause as well. Yep. Angry Penguin, you're creating a hotspot in the incubator reminded me of when a worker removed 2,000 hot flasks from an autoclave and put in a walk-in incubator instead of cooling under laminar flow first. Yeah, that would make them really hot, right? Oh my gosh. Did I mention HIV? Yes, I did in that little talk. Yeah, I did. The incubator got really hot. Vincent, I recommend you check out the podcast on Huberman recently guest series with Dr. Matthew Walkler, sleep researcher. Proper sleep is the foundation of good health. I don't sleep well. I will look at it to see if it can get some hints for sleeping well. But yes, I have a problem with sleeping. So let's see, Huberman, very popular podcast, all self-help stuff, right? Dr. Walker. Sleep. Okay. I'll try and listen to that. Thank you very much. Tropical island. The UV light. Yeah. From the sun will eventually get to your DNA. You're absolutely right. I was just trying to think of a pristine environment, but I guess there isn't any. You could use a lot of sunblock, but where would you get it, right? I don't know. All right, so Portia says, don't just eat well and exercise, which I do. You have to sleep well, which I don't. Visto says, do retroviruses get into sperm or egg cells? So, yeah, if they get into the germline, then yes, they will be in the DNA of sperm and eggs, so they will be passed on to offspring. So that's why it's called an endogenous retrovirus, because it is in the germline and can be passed on. Yeah. Doreen says, I saw a documentary once. Better cancer treatment may lie in focus on categorization of cancer by type rather than primary location. I don't know much about cancers. And yeah, it's true that locations are often a certain type of cancer, but there are some commonalities as well, but I really don't know at all. This is not my wheelhouse, so someone else needs to answer that. Lori just read Breathless by David Quammen. Yes, it's very good. And we interviewed David on Twiv shortly after the book was published, so you should check that out. And he talks all about how he wrote the book and so forth. It's really good. I like the book. It's good. Do we know why DNA vaccines didn't work and why mRNA vaccines did? I think part of the problem is expression level. So for the DNA vaccine, it's got to get in the nucleus, be transcribed to mRNA. The mRNA has to go out and make protein. And I think that's very inefficient compared to putting a ton of mRNA in and getting it translated directly in the cytoplasm. Yeah. Is there anything really clinically significant worth mentioning on human metapneumovirus? No, it's just that it's one of many viruses that cause common colds, okay? They're relatively mild infections. And you can put that with rhinoviruses and common cold coronas and some adenoviruses, not all. So it's just another one to add to the diagnostic panel. Yeah. It was 5 days from the time my PCP phoned for a course of Paxlovid to the pharmacy till the med was available. Maybe if planning on Paxlovid, then keep it on hand. So we had a letter to the clinical update today to Daniel saying, my physician wouldn't prescribe Paxlovid for travel. And Daniel said, you find another physician because you don't need to be COVID positive to get Paxlovid. You can get it for travel. So you just need to find the right person. Doreen, thank you for your contribution to science communication. Love the mini lectures, clinical update, beyond the noise, and TWIVs. Thank you so much for being a loyal listener. We appreciate it, and we like teaching you. Random question. Public toilet seat liner papers, pointless or purposeful? I think they work. The problem I always had with those is you sit on them and they move. And then you touch the seat. So if you can figure out how to sit on them and be covered with paper, that's good. They protect you from the seat itself. But of course, then you're still going to get aerosols and stuff. So thank you, John, for your contribution to science communication. Donated my weekly 20 to the link on the screen to nonprofit science contributions. So Microbe TV, microbe.tv/contribute. If you donate to Microbe TV, you get a federal U.S. tax deduction because we're a 501c3. I can't answer this one, Stone Cookie, but I'll put it up. If we put a 20-inch box fan with a filtrate 2800 20 by 20 air filter on 4 sides of the hospital bed of a patient with measles, will not be enough to prevent transmission. What are these fans going to do? I'm not sure what direction they're going in. Are they going to pull the respiratory exhale away from the patient or keep it to the patient? Are they going to have HEPA filters in them so they can filter the air? If it's a high turnover of the air, yes, but I'm not an engineer, so I don't know what that should be. But in principle, it could work. It's a play on black and yellow. My name rhymes with black and yellow. Carol says, I eat broccoli sprouts and cruciferous veggies containing sulforaphane, which has been shown to reduce cancer risk. That's absolutely right, Carol. I've read that also. Alan says, the must-read books are Spillover, Breathless, The Premonition, and Tell Me When It's Over. If Michael Warby ever writes one, I'll be the first in line. What if I write a book? Would you buy it, Alan? You don't have to say. Portia, thank you for your support of science communication. I really appreciate it. I checked out your message about sleep, and I will look into that. Yeah. John, Uri Berliner at NPR resigned, including a lab leak rant, several paragraphs long among his grievances. How can we combat bad journalism? Invite them to virology meetings. I don't know. I don't know how to combat it. It's a problem. I ask people all the time how to do this. His grievance is that the lab leak is not getting enough air. It shouldn't. There's no evidence to support it. If there were, sure, we could give it air, but there isn't any. Uri Berliner, goodbye. Don't let the door hit you going out. I do think most journalists have good intentions, but they don't know the nuances of science. You really should listen to TWIV and other similar podcasts. Yeah, they should go to virology meetings, but they'll say, I don't have time to do any of that stuff because I'm too busy cranking out 20 stories a day. Oh, my old internet router gets a stray electron several times a day and resets. That's what it is, stray electrons. A solar event? Yeah, that could be. But it happens now and then, you know? I don't know. I don't get it. An engineer friend described these kinds of glitches being caused by lumpy trons. It's great. Every time you say gag and pull, I imagine some cellular comedy team. That's great. Really good. Oh, he was sure that the pandemic was caused by a lab leak, and powers at MPR suppressed coverage. They were right to suppress coverage of nothing. There's nothing to it, Mr. Berlinger. Oh, my gosh. My gosh. Avian pox is a virus from a mosquito which can bite a young turkey poult. I was wondering if it was a type of cancer, too, or is it just a virus? I don't know that it is a cancer. I'm not aware of it. Stray electron should be a band. It should be. Avian does a, let's see. We're almost done here. I don't think they cause cancers. Avian pox is suspected of having oncogenic properties. Columbiforms often develop skin tumors after dry pox infection. If you know what a columbiform is. So, yeah, it could be. Yep. Let's see. We have so many left here. Sorry, I won't get to them all. You are full of questions. I love it. Oh, Liz got alpha gal. Watch the tick bites for sure. TWIP. Go look at TWIP for an alpha gal episode. Okay. Any update about CMV and HIV? As far as I know, they're enrolling in clinical trials. That was I got his name right too. Let's search TWIV, HIV, HCV. Those not HCV, HCMV. Klaus Freu. I guess that's how you say it. Klaus Freu. Let's see. Our lesson with Vinny. That's cute. Our lesson with Vinny. Can you explain virus mutation principle in polio OPV? Mutation occurs randomly, or is this some favorable mutation? No, they are random. But eventually, the mutation is going to occur in a place that is essential for fitness of the virus. Like the 5' non-coding region will change the Sabin-introduced mutation to its original, and the virus becomes more fit and causes paralysis. So I don't believe there's any specific place. Now, you can prevent that by introducing changes up there that make it hard for it to revert. So you can bias against reversion, but the mutation rate should be even across the genome. Ticks. Is Kathy the only remaining TWIV host that hasn't been a guest on office hours? Let's say we've had Dixon, we've had Alan, we've had Brianne, we've had Angela, we've had Rich, right? Who else is there? Jolene, I asked. She said no. And I haven't asked Kathy. But at some point, maybe, yeah. Will COVE run out of spike changes to evolve? I think there are a lot of potential changes, but whether they all confer fitness isn't really clear. So it's too early to tell, right? All right, I think we ought to wrap up. Even though we have lots of great questions, I think we should wrap up here. Although I could go a few more minutes because I did start late. So let's go 10 more minutes if the moderators don't mind. My train is until 10:48 anyway, so I'm no rush. Let's see. What are the key SARS-CoV-2 genetic markers that indicate increased virulence or transmissibility? And how can this be used to predict virus evolution and guide public health response? There are no key markers. And it's not really about virulence and transmissibility. It's about fitness. Everything comes down to fitness, whether a virus can reproduce to sufficient levels to maintain itself in a host population. And that could be almost in any gene in the viral genome. Fitness can be conferred by immune evasion, the ability to evade antibodies upon coming into a host and get a foothold on replication first. So the spike gene, of course, but there are other genes that can also have an influence. And so I don't think we can do any prediction about evolution and use that to guide public health responses. We need to have effective vaccines, have effective testing policies, and then you don't need to shut everything down. You need to test and then treat people and tell them to stay home, have people mask appropriately, and we don't need to close that. I think closing was a disaster for the world. And so we've learned that appropriate vaccines, antivirals, masking, and testing are all part of a program that can preserve society. Let's see here. I'm going to skip Renatus because I don't know what are the infectious cycles for RSV, influenza, and rhinoviruses. So influenza is a relatively fast one. It's about the peak of virus shedding is 2 or 3 days after inoculation of volunteers. And then the symptoms are gone the virus is basically gone in a week and symptoms begin to subside, but they don't go away because there's an inflammatory phase as well. Rhinoviruses, Amy's going to get me on this one. I'm not sure about rhino and RSV, but they're not far off. They're not going to be very much longer. Now, measles has a longer incubation period, and so 2 weeks or so, and that's distinctly different. Yeah. Even as a complete layperson, I appreciate your contribution to my education. You're welcome. It's my pleasure. How about the New York City news about increase in leptospirosis from rat urine? How can we avoid it? No stoop sitting, no grass picnics in parks, wash hands a lot. Yeah, wash your hands. Don't touch your face until you've washed your hands. I think that makes a big difference. You sit on the grass, you put your hands on the grass. You lean back right on your blanket, you put your hands on the grass. They could be contaminated. Then what do? You rub your eyes, you rub your face. So wash your hands. I think that's a big thing. Yeah, I've heard about this. This is a big deal. And now they want to try and decrease the rat population in New York by making them sterile. Good luck with that. Shouldn't we try to convince farms to require protective eyewear on farms that test positive? I do think so, because those infections can initiate in the eye where there are alpha 2,3 sialic acid receptors. Yeah. So I agree if they don't already do it. I lost my place. Yeah, they're no longer called boosters. I know. It's a remnant of my thinking, but they're just new versions of the vaccine. That's fine. Yep. Ticks cause a lot of diseases. Yeah. Rorby et al. Analysis pretty much nailed the lab leak hypothesis shut. That's absolutely right. And the naysayers say it's fraudulent, and they're wrong because it's not fraudulent. They can't fight it. Why do masks work? Well, a well-fitting mask, right, contains most of your respiratory exhalations. They're droplets. They're caught in the mask. So the droplets have viruses in them, and the droplets get caught in the mask material. The viruses don't because they're not exhaled as single virus particles. They're always in droplets. And similarly, so if you're exhaling viruses, you're not going to infect other people. And similarly, the inhaled air will mainly come through the filter, but some will also come through the sides depending on how tight it is. That's why a tight-fitting N95 is the best. A surgical mask with gaps, it's not going to be as protective. But they work. And tell them that's why surgeons wear masks during surgery, so their breath doesn't infect your open gut. Next, if you don't believe in a mask, tell your surgeons not to wear masks next time. See how that works out. What's the problem with epidemiology? Epidemiology does not explain biological properties of viruses. Epidemiology is an observation of what viruses do in human populations. And not all of that is because of the virus, because it's also because of what people do, and much epidemiology doesn't take that into account. Throughout the pandemic, they were calculating R naughts for new variants without considering what was happening to the people. So that's my problem with epidemiologists. It's great. You want to record the number of cases. You want to track spread. That's great. But don't tell me this is more transmissible or virulent, because you don't have any way of measuring those properties. That's not what epidemiology does. Stay in your lane. Spamlet. I tried various notes apps, but in the end found it's most reliable to create text files and then file them. Yeah, you got to have something that's going to be on all computers. I know, Dropbox or Google Docs and so forth. I don't know. I mean, I have a phone with me all the time, right? But I don't like typing on the phone particularly, especially this little thing, which is now broadcasting this live stream hero of a little crappy SE. I shouldn't say that because now it's going to crash. It's hard to type on. I make a lot of mistakes because the keyboard is small. So I don't really like typing on a phone. The Deceivers Club. That's great. It's really great. Oh, Google virus imaging. I saw there was a question of that, but I don't really know too much about it. Let's go back to it. We could try. I'm going to lose myself. Here he is. Oops, now it went away. Sorry, I lost it. Alex L. Here we are. Tom, I ask because I currently work doing imaging and linear unmixing in biofilm microbiology community. I have to find the original email because I don't understand any of that, and I can't find it. So come back next week, Alex, and I'll try and address it. There's lots of imaging going on, especially looking in cells, that sort of imaging. But I don't know if it's what you want. Yeah, 642 doesn't really help. There's a new chemical rat bait that sterilizes rats. That's what they're trying to use here in New York City. What happens if they get resistant to it? A box fan. A HEPA is not a HEPA until it's certified. So attaching a HEPA to a box is no guarantee the HEPA is not leaking. Yep. Oh, my gosh. TWIV 876, spillover market with Michael Warby is really good. I have a few of his proteges that I'm going to get on TWIV to explain that as well. So that's really cool. Yes, it is truly amazing that my phone is broadcasting this event, isn't it? Holy crap. What a computer. All right, folks. I think that's enough for tonight. I've reached the end of your questions, and I appreciate all of you coming and being patient with our technical glitches. We seem to have them regularly. Mostly we get them solved, right? You just need to, the chat timestamp. Apparently there's a pref that I have to turn on to timestamp the chat. You're right, and I meant to do it. Any of you mods have access to some of those features. You could try it, but I'll see if I can remember. I forgot. Someone did tell me about that, and then it would be easier to find them. Otherwise, you just have to scroll. Anyway, let me thank our intrepid moderators. Andrew, Tom, Les, Steph. Who else was here? Did I get everybody? Yep. I got them. Andrew, Tom, Les, Steph. I'm scrolling up to see if anybody was here earlier. Nope. Thank you all for moderating tonight. Really appreciate it. I can turn timestamps on myself, James. Yes, I know. The problem is, once I'm finished with this live stream, it's going to leave my mind. I'm going to forget about it because I have other things to do. So that's why I'm spreading the word. Thanks to all mods, and thanks to all of you for coming. All 200 of you. We got 200 likes. Thank you so much. We will be here next week, I'm pretty sure. Let's take a look at that. Next week it will be April 24th. Live stream at 8 p.m. I don't know if I'll have a guest. I hope I don't have any troubles, but thanks everyone. See the vertical dots at the top of the chat window. Let's do it right now, live. Timestamps. There they are. Yay! I got to bring up what's here. James. Thank you, James. It was so simple because it's right there. Now we got timestamps. Oh, this is so cool. I like when things work like that. Okay. Thanks, everybody, for coming. Looking forward to seeing you next week. I don't know if we'll have a guest or not, but either way, it's very nice to see you all. Where's my plaque thing that I have to put up? Here it is. Okay. Thanks for coming, everyone. Until next week, be safe. Good night.